Welcome everyone to the open talk on cell and gene therapy. I'm Alessandra Picella and I have the honor to lead the Science and Technology Office of the Embassy of Switzerland in Seoul. We are now giving some time to the participant to join the webinar. In the meantime, I would like to introduce about our office. We belong to a big network. The name is SwissNext, whose mission is to connect the dots in science and technology, innovation, and higher education between Switzerland and more than 25 countries in the world. Today's event falls under the umbrella of the Life Science Initiative, a public-private partnership which was founded based on an MOU between the Swiss Ministry of Economic Affairs, Research and Education and the Korean Ministry of Health in 2016. The two governments and the Korean branches of Swiss companies like Novartis, Roche and Roche Diagnostics contribute to the initiative, aiming to foster R&D cooperation in life science between the two countries and support innovative startups. I'd like now to welcome and introduce the speakers of today, and I will do it in alphabetical order. I kindly ask the speakers to turn on their camera, and once I introduce them, to simply say hello in order to show you to our participants. So in alphabetical order, the first speaker is Dr. Brian Che, who is working on cell therapy and tissue engineering from since more than 15 years at INA University. His research covers from the basic science on stem cells and translational research to develop commercial products treating various incurable diseases. In the meantime, he's also working for Strategic Center or for Regenerative Medicine as Vice Director since its establishment in 2011, and for the Council of Advanced Regenerative Medicine as a Secretary General from February 2020, which is commissioned by the Ministry of Health and Welfare in Korea. So please, Dr. Che, say hello to our participants and turn on your camera.
Hello, all. Welcome all participants and audience in this quite exciting webinar today. Thank you for introducing me. Yeah, hello.
Thanks a lot. And the second speaker, again in alphabetical order, is Sandra or Sandy Reyna. She's based in San Francisco. So she led the Neurology Clinical Trials Unit as director and also keeping her appointment as co-director of the Pediatric Motor Disorders Research Program and as a project director of NeuroNext and the Stroke Net. For the National Institute of Health Grant Awards. Since 2005, she focused her research on neuromuscular disorders on spinal muscular atrophy. In 2015, she transitioned to her first industry position at Biogen, and she gained experience in pharmacovigilance, clinical pharmacology, and clinical development. In February 2019, she transitioned to Bluebird Bio to work on gene therapy clinical trials for adrenoleukodystrophy. Currently, she is a Vice President of Global Medical Affairs, Global Therapeutic Area Head of Spinal Muscular Atrophy, aligned with Novartis Gene TX corporate strategy. Please, Sandra, turn on your camera and say hello to our participants. Turn on your mic, please.
Yes. Hi, thank you. Thank you so very much, Alexandra, for the wonderful introduction. And it's a pleasure to be here today. And I look forward to this wonderful meeting we're going to have for the next 2 hours.
Yes, thanks a lot. And Dr. and Professor Orlando Scherer received his master from ETH in Zurich and PhD from Harvard University. He held faculty positions at the University of Zurich and the Stony Brook University before taking up his current position in Ulsan here in Korea as Associate Director at the Center for Genomic Integrity of the Institute for Basic Science and also as Distinguished Professor of Life Sciences Sciences at UNIST. His research combines chemical and biological approaches to study DNA damage, DNA repair, and DNA damage signaling pathways and their relationship to carcinogenesis and anti-tumor therapy. So thanks a lot for to all the speakers to be here. Please, Professor Scherer, turn on your camera and say hello.
Hello, everybody, and thank you very much for the opportunity to contribute to the discussion today. And I'm really looking forward to the two talks.
So I'm gonna just quickly explain how the event is gonna be developed. So there will be first presentation from Dr. Reina and then Dr. Che, and the presentations will be followed by a panel discussion, which is gonna involve also Professor Scherer. And the panel discussion will be moderated by Dr. San Hee Min, my colleague and innovation scout in our team. I kindly ask the audience during the presentations and eventually also during the panel discussion to type their questions in the Q&A session here on Zoom section, sorry, and please also indicate to which speaker your question is addressed, and use instead the chat section just to signal any technical problem. Then I think it's all on my side for the moment, and I kindly ask Dr. Reina to share her screen and start the presentation.
So thank you everyone for your attention today, and I'm really looking forward to having a very active interaction with the group. It's my pleasure today to speak to you on cell and gene therapies and some of the challenges and successes in the development and patient care evaluation of this. So next slide. So what I thought I'd do is, first of all, just really much cover what, how I can summarize the development pathway of Zolgensma and describe the difficulty in developing cell and gene therapies, of course, such as Zolgensma, and then how we manage some of these challenges. I will also cover a little bit of the social value of cell and gene therapy, and I will give you some examples of that. So I will start first with the development pathway of gene therapy. Next slide. So on this slide here, as you can see, we are really trying to show you how far we've come, right? So from the 21st century, it turns out to be an incredibly promising century, because we can see from small molecules to biologic agents, we are now really entering a really exciting era of advanced therapies. Cell and gene therapies are positioned to be promising and have the potential to treat rare cancers and inherited genetic diseases. So in my next slide, I can kind of show you a little bit, some more of that excitement that's been happening. And of course here, you can see that gene therapy has really achieved significant milestones over the last 30 years. So as you can see, follow this timeline, it's been quite challenging, but at the same time, it's brought us so much knowledge from one program to the next, to the point where in 2017, the FDA approved the very first CAR T cell therapy, as well as the first directly administered gene therapy to treat a form of retinal dystrophy. And even closer to that is certainly in 2019, where we have the FDA first approval for a systemic gene therapy to treat SMA, or spinal muscular atrophy. This is, of course, it's been followed now by subsequent approvals, not only in the US, but also in Japan, Europe, and Brazil. As I will discuss later in this presentation, the evaluation of these innovation gene therapies can be quite complex, as we need to consider clinical as well as economic considerations. Next slide. So here, I can tell you, of course, the fact that we can really now have examples of how active gene therapy trials have been and are really still currently underway, right? In trying to treat a monogenic neuronal condition. As you can see, we have, there are a number of ongoing gene therapy trials, and these are a good example of how does it start and where does it end. So we have our preclinical studies in ALS that continue to be there, and of course, and we go from phase 1 all the way through approval in some of these rare diseases. And of course, and now we know that SMA has been one of them in having now been the first gene therapy approved for a rare disease. Next slide. So when we talk about cell and gene therapies, there are mainly two main routes of delivery that we consider, right? On the right side of the slide, you see the cell-based delivery. This is the ex vivo model. And this is when there is a modification of cells in culture followed by cell expansion and injection, right? And an example of that is the Kymriah model. On your left side is a direct delivery. And this is the in vivo model. And this is when there is a direct gene delivery via injection to target tissues. And Zolgensma, or as we also know this product as AVX-101, this is the model of a direct delivery. Next slide. So when it comes to our models here, of knowing that the design elements of the Novartis AAV9 is really our gene therapy platform. We really have, Zolgensma is really designed for target delivery and is designed with a non-replicating AAV9 capsid that has really the ability to cross the blood-brain barrier to transduce target non-dividing motor neuron cells. Right, so there's really key features of this AAV9. First of all, is a single-stranded genome that contains a gene of interest. Its transcriptional regulatory elements, of course, and then the AAV9, which has an inverted terminal repeat. This vector, it has a defective replication, which means it cannot replicate, which makes it a non-pathogenic. And it really persists as extrachromosomal as an episome. So from what you can see here, it's really AAV9 can really penetrate, and this is exactly what we need to target for a motor neuron disease such as SMA. And this work that you see here, we're so proud to have Kevin Faust be working with our team, continues to be there with our gene therapy program, and he really launched the very first discovery that allowed us to understand gene therapy deliveries. As it now we know, it can cross the blood-brain barrier. So this was a one-time study that he was able to show in a mouse model how this could really happen, and it led to what we know now as Zolgensma. Next slide. Okay, so here, so how does Zolgensma work, or gene therapy? For one, gene therapy is really a logical approach for spinal muscular atrophy. Why? Because it's a monogenic deletion or mutation that drives the pathology. So Zolgensma is designed to target, it's a targeted delivery actually, with a non-replicating AAV9 capsid that has the ability again to cross the blood-brain barrier. This allows for transduction of target that is a non-dividing, for motor neuron cells. And so again, so how does this work? Sometimes in very simple terms, this is an intravenously administered medication and is believed to have the effect on spinal muscular atrophy by infecting the motor neurons or myocytes of patients and efficiently expressing human SMA protein or survival motor neuron protein. In addition, the gene expression construct in the Zolgensma exists in the nucleus as episomes, without integrating into the chromosome of the target cells. And the human SMN protein is stably expressed for a long time with a self-complementary DNA and a continuous promoter, which provide rapid activation and expression of the SMN transgene. Next slide. So gene therapy, as an AAV vector development is really complex and sophisticated process, and it has a proprietary process here as outlined in this slide. There are mainly 4 main steps of drug development for gene therapies. For one, we have a transgene development, which is really using a reverse transcription. We create a functional replacement gene to replace that mutated or missing gene. Then we have the AAV vector production, which is a vector that is produced during a rigorous manufacturing process. The viral genes are deleted and replaced with the transgene and the DNA cassette. Then there's a purification process or a purification step. This is the part of the process that allows for production of vectors with high titers, high potency, and high purity. And of course, last but not least, we have the quality assurance, quality control, which this is the process that allows us to ensure that we are delivering products that meet FDA and other regulatory agent guidance and standards. Next slide. So now I'm going to go into what value of cell and gene therapy can provide. Next slide. So there is a potential of elements when we think about value, right? We have to consider some of these. And why is that? Well, anytime we have a new therapy, we have to know what that new therapy is going to be able to achieve. But most certainly also, we have to consider the value and outcome, right? The cost. So there are some parts of this that is going to be humanistic. And in the other part is going to be economic. So on this value wheel of the slide, this is really intended to demonstrate just some of the considerations. I'm not going to go through all of them, but you can really see it's pretty, self-explanatory of some of these value items that would have to, components that have to be part of as we consider what value a new therapy could bring. And ultimately, really the definition of value can be thought as of overall outcomes over the total cost. Next slide. So so that's on the money side, the cost, right? And then when we talk about patients, that's humanistic. So this is really when we think about SMA, when you know what SMA really is and what it does to patients. If a patient is untreated, it leads to infant mortality. As this is the most severe form of the disease, right? And so when it comes to treatment options, children can have a therapy through many. Now, actually, there's other two therapies available. But once that option is there, we can clearly see the value of these therapies because it changes this patient's life. So, I've been talking about SMA. So, what is SMA? Spinal muscular atrophy is a rare disease that is caused by the deletion or mutation, as you can see from this cartoon, of the SMN1 gene or the survival motor neuron gene. This leads to insufficient production of the survival motor neuron protein, which in turn causes weakness and wasting in the voluntary muscles of infants and children, and more rarely in adults. Although the classification is now changing, traditionally there have been 4 types of SMA, type 1, 2, 3, and 4, which type 1 is the most severe form, which is also known as the floppy baby or the non-sitter. Untreated SMA type 1s is the most common genetic cause of infant mortality, with about 85% of these children passing before the age of 2. And so it's very clear to me here from this cartoon that, what an individual who has both copies of SMN1, SMN2, they have a full production of that protein that is vital to all our organs and must have, otherwise these children would not survive if they didn't have that backup copy, which is known as the SMN2. It's a backup copy, but just keep in mind, that's exactly what it is, right? And unfortunately, as you can see here, it's not a perfect copy. It's a faulty copy that produces about 10% of that SMN protein, which is what makes these babies survive. Next slide. So when it comes to disease burden for SMA patients is really quite high. SMA is really associated with an enormous amount of burden, as well as healthcare system cost. They have significant medical expenditures, as well as high resource utilization. And these patients, they get sick, and they get sick pretty fast, literally within hours. They can end up in an intensive care unit, with, starting with a mild fever, and it turns into something major, literally within hours. These children require multidisciplinary care. And that alone will drive up the cost. So you can see here what a type 1 is, which is the weakest type 2, which is also known as a sitter, because they're only able to sit, they never walk. And then the type 3, which are also known as walkers. And although the cost is less, it's not really that much less, right? Still, you're looking at about 100K per year for, to support these children, to keep them as healthy as possible. So my next slide, I'm going to now show you, how to accelerate the development, right, of cell and gene therapy from lab to clinic. So in the next slide, there's a few areas of focus during the development. So what you can see on this slide is that the importance, the importance of consideration when developing a new therapy is, first of all, there are a lot of checkpoints that must be considered. And one of them, that we can start with the market dynamics, right? Here you would need to understand the competitive treatment landscape that you are entering, including pricing, perceptions, et cetera. And not to ignore the epidemiology, right? It's important to understand the overall health status of populations, learn about the disease state, treatment guidelines, standard of care. And of course, then we look also at research and development, what clinical trials are needed to advance this therapy, what outcome measures, safety considerations. And this is a big deal when it comes to any new therapy being considered. And then we have the health technology assessments, right? How do you demonstrate value and pricing? And of course, the cost of manufacturing, this is not to be taken lightly. Building and scaling up production capabilities can be costly and complex processes, as I explained earlier with our gene therapy. And then, of course, the supply chain, this part of development process ensures safe and timely delivery of drug to sites of care. And of course, then we have patient support services. So in my next slide, I can tell you that bringing gene therapy to patients with rare diseases, it's a sense of hope for potentially life-changing therapy, and especially with SMA, families understand exactly what caused SMA in their child, and so therefore, when gene therapy became available, or even the thought of gene therapy, they knew exactly what it would do. It would treat the root cause of the disease, which is the deletion of SMN1 gene. There is a need, there's the same need for patients who suffer rare diseases. It doesn't matter whether they're in thousands or hundreds of patients of the same condition. And then the gene therapy research and development communities can really help work towards lowering the cost of faster and timelines, so that there's more gene therapies that can become available to more patients. So partnerships are critical, and partnerships exist across academic investigators, and also amongst patients and researchers, right, which help us lead the center creation of care guidelines, the newborn screening, which is critical for SMA, for example. So early diagnoses, the sooner you treat, the better these patients do. Trial readiness, that takes a lot of effort and time. And so certainly in order to do that, we have to have some kind of a comparator that we can go back to, which really goes back to natural history studies. In biomarkers, this is a big deal. What can we do? What can we learn about how to best provide prognosis to these families who do suffer from a rare disease, and now there's a therapy available? What are the prognoses for these children once they get treated? And of course, outcome measures, that alone a challenging part of clinical trials, as you can imagine, in trying to find the best outcome measures to measure efficacy. So in my conclusion slide, I'd like to round up by saying that cell and gene therapies certainly offer the potential for short-term and one-off treatment leading to long-term benefits, right, including elements of value that may not be captured in the usual payer value assessment. So Zolgensma has yielded, sustained durability with our patients who have already been treated for over 5 years. It may have, but we still have a lot to learn. We still have to see what happens in the next 5 years, for example. And of course, the cell and gene therapies need to be valued differently to other pharmaceutical products because they are different. They're one of a kind. And the current costing models need to be adapted to ensure rapid patient access to these innovative treatments. So with this, I will close my presentation and give it back to you soon.
Thanks a lot. Thanks a lot, Sandra. And I kindly ask now Dr. Che to start his presentation by sharing his screen. The participants, if there is any question on your side, please
Okay, hi, this is Brian Choi. Can you hear me?
Yes, we can hear you.
Okay, let me start talking about my presentation. I do have quite a number of slides here in my presentation, but in a short time of 20 minutes, I just want to deliver a brief overview about what Korea is doing in the regenerative medicine field, and not going to touch all details in each of slides. Okay, to the next. Okay, my talk consists of 3 parts: market and products, about companies and company alliance, and ecosystem government support. And the market-wise, we first want to touch the not just the Korean market, but Asian market overall. So it's fast growing field in the world, and we are highlighting the potential. Being market in the future in many Asian countries. Best, definitely based on the large amount of populations in this sector. But the Korean market is just still small. We estimate it's less than 1% of the global market. So we highly thinking that we need to go abroad with regenerative medicine products in Korea. But in the commercial development wise, we have already quite a number of products marketed in Korea, not in the global level. You see here, mostly they are based on the somatic cells, autologous ones. There are some allogeneic products, and as you notice here, we do have already 2 immune cell-based therapy to treat cancers early in 2007. And we have first country to give out a market authorization with the adult stem cells in 2011 in number 10 here. It's based on bone marrow MSCs to treat acute myocardial infarction. And in the series, we had 3 additional products using autologous or allogeneic MSCs treating these cancers. So in its number 16, it's a second to the case of US, but without the 8 cases of hematopoietic progenitor cell products, I want to say Korea is number 1 actually in the market products. But we do have quite some limitations as well. Most of products are MSCs, simple culture expanded and somatic cells. And as you see, first approval of gene therapies and CAR T therapies in the middle of this century, a boost of the technology innovations and open the market wide. So we need to catch up with those efforts in Korea as well. So trials are increasing gradually, and I want to say sponsor-initiated trial is overpassed the study level trial. And mostly the trials are using cell therapy. As I mentioned, gene-modified cells or gene therapies are still limited in the clinical trial. But we do have a good number of trials in the immuno-oncology sector as well. But again, no CAR T style therapies are going on yet. And gene therapy in its number between 2007 to 18, the Korea covered just 2% of the trials overall. So we need to boost up the gene therapy sector in the, technology development and commercial efforts in the future as well. So in summary, we are sort of early adapters in the cell therapeutics field in terms of particularly commercialization of products, but we are still behind the technology innovations, particularly the gene modified cells and cell therapies. But you will see in the company activities that we are doing on the field as well. So the number of RN companies, as we estimate in 2019 and 20, is 9 to 68. As you see here, there are 26 uh, companies working on gene therapy, but no clinical trials heavily going on yet, which means most of these companies are recently recent startups and at the early stage of development. We do have again many companies developing a immuno-oncology products and CAR T therapy companies are working, developing a CAR T therapy. We have 14. They are at the R&D stage, but will start the clinical trial in the future, I expect. And in this sector of regenerative medicine, we have many business activities going on between the new startups and pharma companies. And actually in Korea, M&A model is not that common and popular, but we do have many M&A and license out and corporate partnership style business in Korea in the regenerative medicine sector. And particularly in these two cases of Green Cross and ASEAN, they do have a very active the business overseas as well. As I've mentioned before, I'm not going to touch all details on this. And we set up a company alliance named Council for Advanced Regenerative Medicine in 2016. Now we do have 68 companies, including those in therapeutic development and enabling technology and service companies and investment corporation as well. So currently the CARM is led by Busy Ree, a CEO of SCM Life Science, and our mission is definitely to help companies in the field and build up a favorable ecosystem in the regenerative medicine in Korea. And we do have two subcommittee of international network and policy and strategy. As you see here later in the slide, we set up a new legislation on the regenerative medicine last year, and it's enacted at the end of this August, and CARM participated in the process to play an important role as well. So it's what the Policy and Strategy Committee has been done and still going on in our alliance. And we also value the international partnership very much and had an MOU with many related stakeholders overseas, like Forum in Japan, Biotech in Australia, and ARM in the US, and ISCT in during the time. And and it's not always active in the international cooperation, particularly in this COVID pandemic era. But we are trying to communicate, keep talking with them, and trying to find a chance to do something for now. Lastly, about the ecosystem and government, as I told you, we set up a new legislation in the field, and just like the new laws in the other countries, we do have two pillars of promotion and safety, safe use of regenerative medicine. So in the case of the regulatory process, MFDS, the regulatory agency in Korea, used to govern both of the commercial track and study level trials together. And in under this new Regenerative Medicine Act, we separated in two different the program, and you can simply think advanced regenerative medicine is about the research level trial of regenerative medicine. And then ABP is advanced biopharmaceutical as a trial for commercial purpose. And in this part of ABP, MFDS set up reforming, upgrade the regulatory guidelines and rules, and try to make the fast track and conditional approvals, those the innovative reform in place in more robust way. And I want to also already set up the Regulatory Science Center to provide a persistent innovation in the field. And in the case of regenerative medicine, clinical study of regenerative medicine, we adapt not exact. It's a simple, similar to that of the Japan style. I'm going to show you here in the next slide. In the ARM chapter, we evaluate the each of therapies applied based on their risk factor level. And through the series of committee review, we allow the early access of those innovative therapy to patients in need in more affordable way. We also set up the safety management system and data collection and analysis as well. So based on the new act on the advanced regenerative medicine and ABP, firstly we are aiming at the upgrade of regulatory reform in this uh, uh, fast-growing and very promising industry of regenerative medicine to facilitate the uh, patient access of innovative therapy, of course, and also promote the commercialization and industries. And in the promotion part, we um, are for now working on the 5-year master plan about how to keep reform the regulations and policies, and how much of money the Korean government will invest in the field, on what field, and how to encourage industries and academia to make their endeavor to the technology innovation and commercialization and so on. And also we are planning to communicate with many other stakeholders, and particularly patient advocacy in the field. I'm just gonna skip these numbers and we get in the number. So it's almost the last slide, I think. Again, uh, Korea still has some many limitations, and the regenerative medicine is fast growing and very diverse. So it's very challenging to deal with all those uh, uh, fast moving technologies and ecosystems at a time. But we do have ambition to lead the regenerative medicine industry, at least in the Asian sector, and want to be a hub in the North Asia district. So we always welcome any potential collaboration with other overseas companies and institutions, and I want to have many chance to talk with you in the future. Thank you. That's it.
Thank you very much, Dr. Reina and Professor Choi, for your wonderful presentation. Hello, everyone. My name is Sunny Min, and I have the honor to moderate this discussion. I'd like to welcome Professor Orlando Scherer as our panel, joining Dr. Reina and Professor Choi. Hello, Professor Scherer. I believe you will provide us great insights in this topic as a basic science researcher. Great to have you here.
Pleasure to be here.
Me too.
Okay, I see Sandy. Okay, great. So let's dive in. Dr. Reina, for spinal muscular atrophy, there are other therapies recently developed, such as pyridazine derivatives, and one is by Novartis, Brenaplam, and there's Risdiplam by Genentech. I understand that Zolgensma replaces the missing or non-functional SMN1 gene, whereas the others target SMN2 gene. How does Zolgensma compare to these other options clinically?
All right, so can you hear me okay?
Yep.
Yeah.
Good. All right. So thank you for that question. And we get that question a lot, mainly because I think that it's very easy to think about treating SMA and treating the cause of the disease, right? So if you recall from my presentation, I showed you what an SMN1 gene present and absent would do. So normal, patients who are not affected by this mutation obviously have both copies working, functioning perfectly fine. Patients who have SMA have a deleted or have an X on the SMN1 gene. And it leaves that backup copy that I mentioned to you, which is the SMN2 gene. So the difference really is where is the target? And the target for gene therapy is to create that transgene of the SMN1 gene, which is now being really created to act as a gene and be able to now produce a continuous and sustained production of SMN protein. So that's really the difference, right? One is a genetic approach, where the others are really targeting that backup gene, which is the SMN2 gene. And this can be done, as a splicing modifier, which are both, risdiplam and nusinersen. And so therefore, how these work is they really work in targeting that to try to overexpress that faulty gene to produce more survival motor neuron protein. So if you remember when my presentation, I also mentioned that although we have that backup copy, it's a faulty copy, right? It's defective. And so it produces only a very minimal amount of SMN protein. What these therapies do, both risdiplam and nusinersen, is they target that gene and it allows for overexpression of that SMN2 copy gene. So one variant that I haven't mentioned to you is that each patient will have a backup copy, but it also has different numbers of that backup copy. So the least number that you have it seems to correlate with the more severe form of the disease. So for example, I said non-sitters or floppy babies, which are the weakest type, they are the ones who usually have 2 copies of this SMA2 gene, which again, it allows them to have some function, right? But still very, very weak babies. And so it's correlated with type 1. 3 copies of the SMA2 gene usually brings out the phenotype of a sitter, so a baby that never walks, but it's able to sit, or a walker. And so again, that also is another variant which influences on the presentation of these patients. And then you have the adult form. And the adult form can be patients who have the mutation, but may have about 4 copies of that SMA2 backup gene. And so there's a huge difference between gene therapy and ASOs or RNA formulated treatments.
Are they shown clinical significance?
Okay, so the clinical significance, very good. When you ask for clinical significance, we can focus on what data is being produced through our clinical trials, right? Mm-hmm. So the trials done in all these in these 3 therapies have shown improvement, and do they compare? Well, you can imagine, we all sit very tight and protect exactly what our data shows, and consistently our data shows for gene therapy that patients who are treated quite early, which has been shown in our young babies, right? Known as we have the START trial and we have the STRIVE trial, which are young infants that were treated before the age of 6 months. These babies have been able to achieve motor milestones that a floppy baby or type 1 baby would have never been able to do so under the natural history of the disease. And so now they have become sitters, they have become walkers, they're speaking, they're breathing without any respiratory support, which usually is part of the care of these babies, because they get incredibly weak and they can't breathe on their own. So eventually they end up being on a respiratory support for the most part of their lives. And so when it comes to comparison between the 2, I can tell you that our data shows that our patients who have been treated with Zolgensma have not required or have not needed to be placed on respiratory support as if they would have without the therapy. And you can say that the comparison between nusinersen and risdiplam is that patients who have been treated with nusinersen, depending on what age they were treated, that's sort of the effect that they did receive. So nusinersen did have patients who were treated early, such as pre-symptomatic. They also had patients that were treated already, pretty progressed in the disease, and they responded to nusinersen at different levels. But again, the data really is quite different between the 2 when it comes to durability and when it comes to is a one-time dose versus every 4 months. You have to replenish that expression of the survival motor neuron, because the life expectancy of that medication goes away very quickly, right? So every 4 months, nusinersen has to be redosed, and risdiplam, they have to take it daily.
Mm-hmm. Can I ask something here? So you said that the AAV9 delivery goes into the mutant neuron. It's expressed as an exosome. So is that stable indefinitely? Is that expressed indefinitely as far as you know so far in those neurons?
As far as we know, that's what we are expecting to see, right? So only time will tell. I can tell you right now that our data continues to show durability of our patients, even after 5 years of being treated with Zolgensma. Mm-hmm. So they continue to gain motor milestones.
Yeah. Do you have a way to, I guess you look at the milestones and the clinical milestones, do you have a way of analyzing the patient neurons to see what their status is? Or that's probably difficult, I would imagine, right?
Well, no, not really. So again, and I have to be very transparent. I have sort of both stories of the whole process, because I worked in both companies, right? So I know the nusinersen product pretty well, and now I'm working in gene therapy. But overall, the neurology behind this is that you can evaluate and you can screen and you can test your patients to see how well they're doing based on motor neuron health. And how do you do that? You can do that through electrophysiological testing, right? So you can do a CMAP test, right, which measures the health of the motor neuron. You can also do a motor unit, number estimation, which is another electrophysiological test, which tells you how many motor neurons are still there and alive. And so you can certainly do that, but you have,
So you
again,
can
you
do
have to
that
do
in
it
the
from the very
patient.
beginning.
You can do that in the patient.
Yes,
Yeah.
absolutely. Okay. It's a clinical evaluation. So physiological tests can be done to capture that change, which some studies have shown that patients came in with a very low CMAP or immunity, and then after treatment, obviously those numbers went higher, which means that they have a better motor neuron connection to the muscle, so they're getting better.
And so the patient, all this. Go ahead.
Thank you. You said the therapy when treated to patient lasts like up to 4 to 6 months, and you have to re-administer the therapy again, right?
Not for gene therapy.
Not for gene therapy.
No, gene therapy is one-time intravenous injection that is administered over an hour through it's IV infusion, right?
Okay,
and that's a
we have
one-time
already,
therapy.
yeah, quite a number of examples with gene therapy with a good outcome in clinics already. But as a scientist myself, always wondered how the gene therapy, gene modified cells survive for a long time in vivo. So do you have how long time follow the results
So
in your case?
Yeah, right in our case, right now we have over 5 years. We have over 5 years right now of patients who were part of the first trials. We're continuing to monitor them in a long-term follow-up, and they have continued to show stability. They have continued to show gaining of other motor milestones. So I think it's important for us to really know how significant this is for these families, right? Because when you look at these babies and you realize how weak they are and how sick really they are, and fragile, and now you have a whole different phenotype of a baby with a 2 copy, right? A 2 copy SMN2 gene. It's really almost night and day. We still have a lot to learn. This is gene therapy. We're the first ones to do this.
But
Right?
it's
So
amazing
it's over
that
5 years.
administered cells survive more than 5 years and functioning in patients. It's great.
Do you have an idea what percentage of cells you're able to target of the motor neurons, or ask it differently, what percentage of cells do you need to target to have the desired therapeutic effect?
So that's a very good question, actually, because when you think about what survival motor neuron protein would be in a patient like you, me, any other child who does not have SMA, you need about anywhere from 50 to 70% presence of SMA protein to be okay. You don't have to have 100%, right? And as you get older, so it's very high at the very beginning. So intrautero, you have a very high level of SMN protein. And as you grow, as you are born, you have still that high number of SMN protein or percentage. But as you get older, that diminishes. So what we know now is, of course, how will this continue to be present in these babies as they get older? It's only time will tell. But as it is, there's no way that we know exactly the percentage that's being produced. What we know is that there is a continuous and durable and sustained expression of SMN protein through this transgene. What we also know is the biodistribution. Unfortunately, through the loss of 2 babies during the trial, we were able to get autopsy tissue, which showed expression in the key areas, right? So spinal cord, brain, pretty much in many organs, SMN protein was present.
So when you did preclinical experiments on animal, be it mice or I don't know what other animals were used.
Mice.
Mice. Do you see, is the distribution comparable to what you see in humans? So is it a good predictor when you look at the mouse, what may happen in humans?
Yes, it is. And so it's the same for as well as the monkey, in a large animal.
Yeah. Okay. All right.
Okay.
So if I'm now,
I'm sorry. Go ahead.
so if I'm now a clinician and I'm referred an SMA patient, so I have the different options, right? I have the gene therapy, which I assume is a lot more expensive, probably, compared to some of the other therapies. So what are my I mean, of course, you would promote the gene therapy, but so what are my considerations as a clinician? Which therapy would I choose, or what would be, what would I take into consideration?
Yeah, Orlando, what I like to know and what I like to think is that I'm still very much balanced in my head, right? Even though I'm in industry, I'm still very much a patient advocate.
No, sure.
I've been there with these families for so many years. I know what it means to give a diagnosis for SMA. I know what it means to have absolutely no options for these babies. And so if I were in clinic right now, I would still want to be as balanced as possible and consider each patient individually. So if I have a pre-symptomatic baby, and just the way I'm saying it right now, pre-symptomatic baby, this baby's born, I know what I want. I want gene therapy because I'm going to treat the root cause of the disease. If as this baby gets older, catch him early, right? If I can do this, it's one time, and then parents will have a choice. If they feel that they want to do something more, it's their choice to say, we want to see if there's any sequential therapy, which obviously that is an option, and families are doing it as we speak.
So you combine the therapy,
starting.
You use the gene therapy first and then use another, something else later on. That has happened, that people do that also?
Real world evidence is we have real world evidence that's what we're getting. So we have a registry that's collecting that data, and we're able to see that families are choosing to do after they have one or the other, right? If they were nusinersen, we have a lot of nusinersen patients are now on gene therapy. So we're trying to learn. These are options. So to your question, what would I do as a physician? I will lay out the pros and cons of each one, and then families have to choose what works for them. Each family has different needs, and many of them will say, I can't bear of the idea of doing this every 4 months. Others will say, I can't even bear thinking that I could miss a dose or two, the oral administration, right? And so this way, for me, it works better if you just give me one-time therapy and we could be done.
Yeah. Okay.
Okay, well, thank you for that. Let's dive more into the development. For genetic disorders, considering the same delivery system can be used for different diseases, how do you see the streamlining the process for the gene therapy?
Can you repeat that last piece? There was background. I couldn't.
So considering the same vector can be used for different diseases, how do you see the streamlining process for the development of gene therapy?
Well, if the same vector can be used, I mean, I think there's definitely possibilities here, and I know they're being explored as we speak, right? And so, if it's a monogenic disease, it makes it much easier to target, right? So the idea would be to then create that we have that platform. We understand how it works. We understand how this vector has really become somewhat of an ideal vector because of its capabilities, right? Of how it works. So I think the streamlining it would really have to be on where's your target, what organ are you trying to reach, and most importantly, what is the main cause of that disorder.
So along the same line, so with the vector you have now, is this satisfactory to you for targeting neurological disorders, or do you think there's a need to develop better vectors still? And this, if so, is Novartis doing that?
Yeah. No, so right now we're very confident in the vector that we're currently using because again of what we've been able to see, right? As you can imagine, when it comes to side effects, we're still learning, how it's done and why it's done, and how it's managed. So far, we are continuing to work with the same vector as we explore other opportunities.
The therapeutic, I have another question. The therapeutic benefits of gene therapy looks remarkable. It's, we can say, innovation going on in the future as well. But there are still many hurdles as well, technical hurdles and operational hurdles in clinics. And one of them is the cost issue, right? High price tag of gene therapies matters all around the world, even the richest country as well. So they say manufacturing innovation, automation, mass production, and those things could solve out the price issue. I mean, cost of production to some extent, but I think it's still too early to think so. So I want to ask you if how much impact the manufacturing, the innovation will on the price of the product, and how much we will be able to reduce it in what time period in the future.
Well, that's a loaded question, right? So, so you have a lot of components there. I have a lot of components. So
yeah, right.
you, yeah, I think when it comes to pricing, not everybody loves to talk about money, but it's a fact of life, right? And when you speak to families about the cost of these treatments, it's very hard for them to wrap their heads around the price. But when you think of the process, and like I explained in my presentation of the complexities of making sure that this transgene is going to do exactly what you're creating it to do, you're designing it to do. It is incredibly complicated to do that. So a lot of high technology, a lot of trial and error, and then the quality of life. So then you have to compensate for what it would mean for these patients to not be treated, and then what it would mean for patients to be treated with something different than gene therapy. And when you do that and you add all that up,
yeah, I definitely
there
agree
is
with
a formula,
your
right?
opinion. Yeah, I understand the cost benefit concerns as well. But to make my question simple, from the manufacturing innovation, how much percentage of production cost we will be able to down in the future?
I can tell you that. I'm not,
Okay.
I'm not the person, to be very honest with you.
Okay, no
I wouldn't be able
problem.
to,
Thank you.
I couldn't tell you that information. Yeah, I don't even know it, so I wouldn't be able to help you with that.
Okay.
So do you know how many, you probably know how many patients have been treated already with this therapy?
Oh, absolutely.
US or globally?
So US and globally, right? So we have over 600 patients that have been treated with Zolgensma. And that includes clinical trials, it includes some, managed access program patients and real world evidence patients. And the numbers keep rising, right? So this is the number I'm giving you right now, but I know it's going to be different probably by the end of the month.
I mean, that explains the high cost too, right? Because for each monogenetic disorder, you have a very limited number of patients, but you still have similar development costs as if you make a drug for many more people. So of course that explains some of why this is so expensive.
Yeah. Which is, I think it brings another part B to your question is, how difficult is it to design a trial for a rare disease in patient access? It is very difficult. to try to capture as much as possible within that trial design, right? So that you can have a pretty comprehensive data set. When it comes to SMA, I know, and I still remember, thinking about creating my own trials when I was in academia. And even back then, 10, 15 years ago, when I had absolutely nothing, it was natural history studies, right? So creating natural history study, what kind of data do I need to capture? What do I need to understand? So the natural history database has become critical for the SMA, progress that we've made, because it allowed us to understand how the disease would progress, how quickly it could progress, and then how do we capture change? So it brings another complexity to the design, which is what scales do you use to capture this change? And it's taken over 10 years to even find a motor function scale that is sensitive enough that is used and applied in neuromuscular disease patients. So for example, when we're as you probably already know some of this data, when you're trying to see a floppy baby who would be a floppy baby in any change that they have, how are you measuring their evolution, right? How are you measuring their development? So you have to have a scale that can do that. Nicely enough, these babies are really responding to treatment. That the scales that were created for weak babies, they're surpassing those scales. So then it brought another challenge into the trial, where now you're using scales that would capture normal, growth development in children not affected with SMA. So now we're not really comparing apples to apples anymore, because now you're needing to account for, change, but knowing that this baby still has SMA, and the change in their growth is very similar to non-affected babies, very much age appropriate, but is not exactly as you would expect it to be, right? So the bar has been raised incredibly high when it comes to capturing change in clinical trials for SMA. So when you start, it's great because you're setting the rules, but then as you go, the bar keeps getting higher and higher and higher. And the expectations for families are also something you have to manage, right? Expectations also become much higher.
So when you start with a clinical trial, you have the phase 1 for safety evaluation. So is that done on healthy people, even when you have such a specialized treatment?
Right. So for gene therapy, we could not do healthy patients. So it had to be done in
It's
patients
same with the cell
who were
therapy.
affected.
They
Cell
mostly
therapy is
start
the
with the
same thing.
patients from the phase 1.
Yeah.
So even the phase 1 is not an objective phase 1, if you will, but it's really for the people who will
Phase
be
1A,
receiving that therapy.
1 and 2A, yeah.
Yeah, that makes
say.
sense. I mean,
Yeah.
is the patients are so rare, right? That makes a lot of sense.
Right.
Sure.
Yeah. So Professor Che,
Chair, I have a
maybe.
question for you.
Oh yeah. Okay. Go ahead.
No, I just have a question for you. It's a very general one. Do you see any challenges or hurdles in translating basic science research into clinical ones? Into clinical
I
use?
think the scale of doing things is a lot bigger, of course, right? So we will work on a small thing. We have a model system, perhaps, and we have the development. So I think to translate that into a bigger thing is always a challenge. And the way most people would solve it is by either starting a startup company to promote that product or develop that product further into a more clinical realm, or then partner with a bigger company, be it Novartis. Company like Novartis may be coming into the game a little later when it's a bit further along in development. But so I think I think we just think about things very differently, right? Most of the people in academia, and so I think it will be important then to find a partner organization. Could be a government laboratory, it could be a company, or start a startup to do that more effectively.
I think there are more gaps in cell therapy, cell and gene therapy sector in particular, because cells are changing in their characteristics and pattern abilities during the the manufacturing process, and they also change their fate and characteristics inside of our body. So there are many uncertainties still there. So for now, I think we are sort of compromising on those issues with the regulatory bodies to get an approval, because nobody can estimate all those things from the beginning, and it's still underway to dig in those details.
So what are
Professor
some of
Choi,
the
you
Oh, I was going to say,
no,
I
go
have
ahead.
a question. I have a question for Dr. Choi. So can you tell me a little bit about what do you think is the perception of gene therapy in Korea?
I was going to ask the
We
same thing.
are,
Yeah.
as I was showing you in my slide, still at the early stage, and many companies, particularly in the immuno-oncology sector, many companies are there, and many of Korean companies understand that we cannot just compete inside. They are looking for chances to go outside, and partnership with many overseas companies and institutes as well. So I think it's very positive in the future. It may take some time, but many in the technology wise, many Korean scientists have good technology as well, but they didn't know how to deliver, I mean, how to develop the technology to products. So you must understand that it's not just a simple way of translation. There are many things to consider to make it true as a product, right? We haven't actually we haven't those all those experiences in Korea yet, but we are accumulating those and learning faster from the experience of the leading countries. So we will catch up with those parts as well. Still, Korea is a small company, and we cannot be a big good match either. But we will find our own niche and space to build up our territory in the future.
So have any Korean patients been treated with gene therapy products that have been developed in other countries?
Sure, we do have we used to have a cell modified gene therapy, but it was canceled unfortunately. So I have no gene therapy products for now, and we have a couple of gene therapy companies developing a product. I mean, they have done clinical trials in the US already, two companies. They are actually staggering a bit these days, but they are, I mean, we do have some companies and examples of a clinical trial in Korea and in the outside as well.
Yeah.
And so for the companies developing cell therapy products or the immunocancer products, are those in Korea, are those mainly smaller companies, or are they part of bigger companies, or they associate with bigger companies?
Mostly small, but some are sort of a big company, big pharma in Korea, like Green Cross. You must have seen in my list the one of companies developed a the immune cell therapy treating hepatocellular carcinoma is a Green Cross. Actually, I wrote it InnoCell, but I think I have to change it because Green Cross bought InnoCell years earlier before. So it's now a product of InnoCell, I mean Green Cross. So many pharma companies are interested in this sector as well. In the Calm Alliance, most companies are small startups, but we do have Samsung Biologics and LG Calm and Daewon Pharmaceuticals. So we do have some big companies in the alliance and in the sector in Korea as well.
Professor Choi, go ahead.
Oh, sorry. I was going to say, so what are your thoughts on once we jump through those hurdles and you're able to get a therapy, what do you think are going to be some of your challenges to have that therapy, be accessible to patients, like getting it to the market?
The hurdles, you mean, in the future?
Yeah, in the future.
Once many of those current hurdles are solved, your question is, what do I expect? Any further hurdles, right?
Well, once you get that therapy approved,
Okay, okay.
what yeah, what are the hurdles you still have to jump through? Because it's not, getting it approved and getting it, this is so excited and very excited to do so,
Okay, okay.
but then getting into
Uh,
patients, you still have
yes,
other
yes, right.
challenges.
Definitely the price issue. So it's the reason why I have had a question about the manufacturing innovation, how much it will down the price in the future, how long it will take to make it sort of a affordable price that we can have. So particularly in Korea, we do have a nationwide health insurance system, and
Yeah,
we do have private health insurance program as well. But the size is not that big yet. So we do have a good insurance program, but the price tag of cell and gene therapy is beyond the capability of a current program. So we, just like the other countries in the Western field, Korea also needs to deal with this issue, and we need to actually more communication among stakeholders, and that's the first one. But another opinion of me is about the um, uh, uh, how to make it personalized. It's not
hmm
exactly about the patient access, but even the chemical drugs and biological drugs, there are, I mean, big variations from patient to patient, right? So we cannot actually have our best benefit with a single therapy to all target patients. So getting to know more about the patient to patient variation, and to set up the best way to find the how to personalize cell and gene therapy to patient. It also related with the cost, manufacturing cost as well. So it will be the next hurdle we have to deal with in the future.
yeah.
Professor Choi, if we can go back to the hurdles again,
Okay.
you went over the Korean regulation models, and that you mentioned that the part of the structure or process is similar to that of Japan. Are there different models in other countries like Europe or the US?
Europe, and I think Sandra knows much better than me about the Europe prime program, prime process, and RMAT designation. Actually, those are not exactly same as the Japanese style, but they adapt. They try to allow the flexibility in the regulatory process, and allow the more patient access of those innovative therapy. In the sense, they do have a similar concept, and there used to be a hospital exemption in Europe as well. I don't know if it's still working very well, but they were, particularly the case of Japan. They allow the patient application of regenerative medicine uh, similar to the style of medical practice, right? Not exactly. They do have a two process for the clinical study style. I mean, medical practice style application of cell and not gene therapy, cell and immune cells and stem cells, and commercial track allowing the conditional approval. In this sense, we have a similar type of two pillar in the process. So I know many Asian countries are considering similar models, like India trying to adapt the Japanese scheme in their regulatory process, and Taiwan also adapted Japan style and a bit modified it. They already set up a medical technology style treatment in Taiwan, and they are talking about dealing with a commercial track as well. Not yet enacted, but
Okay,
so
thank
it's
you.
like a
Second to
global
the last.
trend. Yeah,
Okay,
yeah, yeah.
thank you.
Go ahead.
Second to the last question before we wrap up. This is a question from the audience. Question to Dr. Reina. Um, you had focused mostly on gene therapy. Can you comment a little more on Novartis' approach to cell therapy, and in particular, any initiatives related to Korea?
Well, I can tell you that it's certainly, that's part of our portfolio. Right now, our focus is gene therapy. So that's really all I can relate to you right now. I'm not necessarily part of the cell, the cell development of that program.
Okay, all right. Last question. This is for everyone. And this question is, well, I mean, many of them work, but this question is from Professor Sher. Where do you see the long-term future for genetic medicine? Can you share your perspectives for the cell and gene therapy, antisense siRNA, and CRISPR-mediated gene editing?
Dr. Choi, do you want to go
Anyone?
first?
Oh, okay. Definitely, it will become true in the future. But how long, how fast it will, I have no exact idea. So there are still many hurdles with particularly the CRISPR technology. But I want to mention that even before we do have a commercial product in the market, we could utilize them. I mean, we have treated them to patient, right? So the first trial, there are already many clinical trials using CRISPR technology as well. And there are already a couple of products using siRNA in the US and in Japan as well. So it's already happening, right? But what I mean is technology maturation and widespread of those technologies in the market will take some time, and there are more rooms for technology improvement as well. But it's very, very diverse and powerful, and will be one of the lead, right, in the future. And there must be a more converging of technologies with those ones and AIs and robotics and so on. I envision those are the future the images in the biomedical sector. Yeah.
Yeah. And from my end, I can tell you again, going back to even 20 years ago, right, when the very first gene therapy was even intended to be a therapy and tried, and we learned the hard way, right? So unfortunately, gene therapy started somewhat with a very sad story and a very sad experience, but that is what science is. In the more we think about it, the more we realize we are flying this plane and building it at the same time. We are being incredibly cautious in trying to figure out, what would be the best method and what would be the best, route to learn from our previous experiences. And so I think moving forward, we are all on the right track. The excitement is there. There's certainly, so much has happened even in the last 5 years. Rare diseases are getting more and more targeted by industry, which is great because that really allows for that growth and development of rare disease management, right? And then the target. So when it comes to the scientific side of what's really happening in these incredible therapies that are coming our way, you think about CAR T cell, development, how that has just grown so quickly, and it's growing, but it continues to do so. But there's also challenges, and we learn from those challenges. And so in the next 5 years, 10 years, I really think that we are going to see a different side of medicine. There's going to be a lot more, individualized medicine, because even for SMA, to an earlier question that I had, what would you do now with the 3 therapies available? It is becoming an individualized way of treating your SMA patients, right? It will be the same when it comes to any other rare disease, and there's so many of them. We have ALS, we have Rett syndrome, Huntington's disease. We have so many other, neuromuscular disorders that will eventually require that type of individualized therapy. So as I think of other, possibilities, there is such a thing as the next frontiers, right? And when you look at CAR T cell development, you know that there's an off the shelf type of cancer therapies that are being now designated, and these are now known as the next frontier because they are becoming unique. Cancer alone is just a fertile ground for science because every patient is very much individual. Every patient will react differently. And it's really no different when it comes to rare diseases. So do I envision that medicine will change in the next 5 years? Yes. Just alone in this past 5 years, right? So from 2007 to now, when the very first standard of care guidelines were written for SMA, and where we are now in 2018, and it was an updated standard of care guideline. And now we have to update those because now in 2020, we have 3 therapies available. So that just tells you the speed that we're going and heading forward with possibilities of therapies as the science continues to grow and continues to become stronger because of so many groups doing different types of work. And that collaboration is so critical because we all learn from each other. So I really see an amazing future when it comes to new therapies and the development of them.
Professor Scherer?
Yeah,
I've not.
Yes. I think I think it'll be very interesting to see how these targeted gene repair CRISPR approaches develop in the future. I think that's something we don't know much about yet. How to use it clinically, how safe it is to use clinically, what is ethical to use clinically. But of course, in theory, that is the best way of doing it, right? To repair a cell in the body rather than supplement it somehow. So I think it's a very long way to go, but it will be interesting to see how that may develop in the future.
Mm-hmm. Thank you. Well, thank you all 3 of you for your time and expert knowledge. I believe this concludes our panel discussion.
Yeah,
thank you so very much.
I also would like to thank all the speakers because this kind of panel discussion is a kind of a dream panel for who organizes this kind of events, because it has been really spontaneous, and we really appreciated all of you to have exchange during the panel. So thanks a lot for this active, vibrant discussion. Thanks also to Sunny, who prepared the moderation, and even if actually a role today, was really like to give you space for the discussion. And I'd like also to thank all the contributors to this life science initiative. First of all, the Korean Ministry of Health and Welfare, the State Secretariat for Education, Research and Innovation in Switzerland, the Korean branches of Novartis, Roche, and Roche Diagnostics for their support, which makes generally these events offline, and now this year, because of COVID pandemic, online possible. But also thanks to the co-organizers from the Korea Health Industry Development Institute, KIDI, for their cooperation and coordination of the activities. Thanks all the participants for having been here, like listening to the presentation, and hoping to meet all of you in the future in person and not just virtually. The next appointments for us under the Life Science Initiative is going to be on Tuesday 27th, where we will focus on the personalized medicine. And now I'd like to ask all the speakers to smile because we're going to take a picture as a memory of this virtual meeting. And we are going to also upload on our LinkedIn the recording of this event for who couldn't attend. And we also ask, as we have been asked from participants, to the speakers if they allow to share the presentation. And eventually we will share with the participants. Thanks a lot, and smile, please.
Okay.
Thank you.
Done. Okay.
Thank you.
Thanks a lot,
All right.
all, and talk
Pleasure
to
to
you
meet
soon.
everybody.
Bye.
Thank you.
Bye-bye.
Bye-bye.